we're going to transition now into our abstract selected talks. So first up is Yixing Han from the National Human Genome Research Institute. It's just a big button here.
Good morning, everyone. I'm Yixing Han from NHGRI, the Center for Precision Health Research. Our group is focusing on the complex childhood diseases. We are trying to use genomic approach to understand those complex diseases. So today, I'm so honored and happy to share with you one of our project focused on the severe acute malnutrition used across AnVIL and the Terra system. So when we talk about the malnutrition, there are multiple forms, include the under malnutrition and over malnutrition. So usually the obesity is acquire more focus, while the undernutrition, usually we call malnutrition, is also causing severe consequences. So globally, there 1 in 3 people has malnutrition suffering, and it causes measurable adverse event on the whole body system, like a neuron system, immune system, and so on. And the malnutrition instance or prevalence is disproportionately worldwide. So, for example, severe acute nutrition, in short, we call it SAM, and it affects about over 17 million children under the age of 5 at any given time. And it's also causing 11% of the mortality with young children in Africa. And in terms of the geography distribution, sub-Sahara African regions and the southern and the central Asia is most intense SAM patients. In terms of the SAM forms, there are 2 types of the SAM in childhood populations. So on the right side is ademptus SAM. The feature of that type is with ademptus, when we push down the when we push the finger to the legs or feet, it's hard to recover very soon. And when you image the malnutrition or hungry children in the Africans, the skinny ones are usually the popular formation, and actually that's a healthier form called the non-ademptus SAM or AE SAM. So those 2 types of SAM developed under the same environmental conditions and with same genetic background. And so far, the treatment in these 2 forms are both give them more nutrition and has no differentiation in the clinical side. So overall, the etiology of the ESAM, or we also call it kwashiorkor, and the NESAM usually calls the marasmus. So those 2 types, we do not understand yet why they developed those 2 types. In a smaller, like a few individuals participated study, the one carbon metabolism pathway is involved in the SAM development, and over the meiosis, the DNA methylation turns to be a factor or consequence that the ESAM has a hypomethylation and the NESAM has a higher percentage of the methylation. And based on those study, we have been worked on 2 cohorts recruited from Jamaica and Malawi with our collaborators. And those buccal swab samples and blood samples, we go through the Illumina human methylation array, and we analyzed over 485,000 of CPT locus to see the hypomethylation status. And based on the 2 cohorts and their stage of the SAM development, we identify over 160 methylation sites. So in the circle, in the circle map, the blue labeled ones are the hypomethylated regions, and the orange are the hypermethylated regions. So we can clearly see the feature that the differentially methylated region is correlated with the ESAM. And together with the gene expression data, we also locate those differentially methylated loci to the pathway, and those differentially methylated loci includes in the gene implicated in the crusher-core phenotypes, and also in the nutrition and metabolic trees. So overall, we sum this progress into a molecular framework. It's a general framework for the SAM and ESAM, for the ESAM and NESAM development. So those methylation focus or centered by the 1 carbon metabolism, 1CM pathways. And with the progress of our recruitment of the patients, we also ask the question, other than the epigenomic epigenome level differentiation, how about the genome level difference? So based on the methylation research and the larger recruitment, larger patients cohort, for this one, we go up to like more than 700 patients. We did the microarray focus on the 1CM associated loci, manually accumulated from the pathway databases. And from the methylation-wise, from pathway-wise association analysis, we identify some of the OCM pathway-related genes. They are standing out from the association study, like the GA, BBR2, the PRIC, LE2. Those are also involved in the 1CM pathway genes, and the cumulative association of the SNP at the 1CM loci is also standing out from random accumulative study. Also, since those 2 cohorts are from Jamaica and Malawi, we also consider if the ancestry background is also one factor related to the ESAM development. From the this graph, we can see the Malawi is much more closer to the all patients cohort Z-score, and the Jamaica is far from there, and we compare to the local ancestry administered result over our entire cohort. We can see the East African ancestry might also play a role in the ESAM development. And how the ancestry factor influence the phenotype development, we are studying that maybe because of maybe through the metabolite variation, and our group member also studied the a pilot experiment on the metabolite variation study, and some of the metabolites are really involved more in the ESAM than NESAM. So in summary, together we have found out that the ESAM is characterized by the hypomethylation of the DNA, and the 1CM is 1CM, and the ancestry genetics is underpin the ESAM development. So those are all our preliminary data for our AnVIL analysis. We try to build up a more comprehensive molecular framework. So based further on the microarray analysis, we also did the whole genome sequencing for those samples, and we are trying to do a much deeper analysis on the genetic variant discovery to see if there are more rare variants or even common variants under the ESAM development. And since we also have the demethylation, the epigenetic information, genomic information, and metabolome and RNA sequencing, the transcriptome data, we are going to work them together to build up a multi-omic integration and use the machine learning to build a model to see if there's any features standing out between the ESAM and NESAM. And this is our planned workflow. So we are going to work on the AnVIL platform to integrate the multi-level omics data together. Since we have over 400 samples, has over 700 samples, and we are also going to integrate with all of us data to do the population and the statistical analysis to see the local and global and local ancestry. Overall, we hope we can find out the ESAM and NESAM molecular signature to inform the diagnosis and clinical treatment. So those are all together a pilot experiment. We are also working on a much larger consortium called the CrushNet. That is also highlight why we are moving to cloud, moving to AnVIL and Terra. This CrushNet consortium is aiming to recruit over 8,000 SAM and SAM patients in the African countries. We are aiming to have the multi-clinical centers to sequence their sample and upload the data in the cloud platform. And together, all the data scientists and bioinformaticians and computational biologists that can work together on the cloud collaboratively. And with that, we are. I'm going to stop here and open to questions. Thanks to the NHGRI, especially the OGDS office, provide us this opportunity. And we are also kind of new, like a couple years on the AnVIL system and the Terra. And there are many challenges, and we are learning a lot every day. And thanks for the AnVIL team for the prestigious support, which is very helpful. Thank you, and I'm open to questions.
Yeah. I'll just say the multi-omics workflow you just showed is incredibly powerful and sophisticated. I guess I'm curious, how far along are you in that process of developing it? I'm curious how that's going for you.
It's different. So all the data are available, and we are working on the whole genome sequencing data analysis currently. We try to focus on the rare virus first, and then go further to the common virus combined with all of our database. Those are in future, and the first arm is working. Yeah, and we try to use the demethylation identified loci, which published a couple years ago, combine that together, and the metabolism data is under the way coming to our server.
Okay, well, yeah. Maybe next year we'll have you back to hear more about it.
Yeah, hopefully next year we'll report more results.
All right, thank you so much.
Thank you.
Okay, next up, Tanguy Rubat du Mérac from Vanderbilt. Why don't you please come on? Just the big button to be.
Okay, I hope everyone can hear me. So nice to be here. My name is Tanguy. I am part of the Music Cognition Lab here at Vanderbilt University Medical Center. And I'm a third-year human genetics program PhD candidate here at the Vanderbilt School of Medicine. Very excited to share with you some of the work I've been doing on the genetic architecture of social music making enjoyment. So a little bit of a different topic, as you can imagine. So before I delve into this topic, I wanted to kind of invite you to think about the last time you perhaps have engaged in some kind of social musical activity, maybe, kind of singing as part of the crowd at church, or, for any kind of religious service, or maybe you just went, with some of your colleagues or friends to a karaoke and started singing along. All of these kind of activities have in common that you're actively engaging in some musical activity, either by singing, playing an instrument. And that has, as we kind of figure out through the literature too, some kind of biological basis and evolutionary rationale for it. So one of the prevailing theories around evolution of music actually tells us that music may have co-evolved as this kind of system for social bonding. So music fosters social bonding, and then as it, population size also increases over history, we can actually witness that music becomes more and more an efficient tool to foster this social connection, maybe more even than other activities such as, sharing food, for example, with others, or maybe laughter. So like everyone likes a good comedy, for example. And so fast forward to our 20th and 21st centuries, we have quite a lot of research actually conducted on social musical activities and interventions that are done to potentially improve some health outcomes from loneliness, from anxiety, depression, your sense of belonging to a community, all these different facets of our health. And so as we see that there's evidence for social musical activities to actually enhance our health in those different domains, some of my colleagues from the Max Planck Institute for Psycholinguistics in the Netherlands actually started diving into, okay, what may be happening biologically that makes us so prone to engage with music, especially in social settings. And so my colleague Giacomo actually started to look into what is the heritability, so how much genetic variation is explained by genetics on social music making and enjoyment. So he looked first at musical enjoyment, so how much joy do you feel when you actually engage in any kind of musical activity, found that musical enjoyment is heritable, all these different facets that you can see here are extracted from a previously validated scale called the Barcelona Music Reward Questionnaire, which includes 5 different domains of musical enjoyment, from emotion evocation to mood regulation, music seeking, sensory motor processes, and the last one that we're going to actually focus on today, social rewards. So the kind of like social enjoyment that you get from musical activities. And so now that we kind of have evidence for social music making to be evolutionary relevant, to have also some positive impact on our health, we can kind of start thinking about, okay, maybe we can personalize care, personalize interventions in a way, and get to a point where we can see, okay, this person is not only willing to take part in this intervention, but they're also very, very much a good fit biologically too, and may benefit more from the intervention than maybe other individuals. And so to kind of get this understanding, we do need to understand more of the biology underlying musicality and how it overlaps potentially with different health outcomes and with different domains of our social-emotional well-being. So the big kind of our overarching goal for this project was really to characterize the genetic basis of social music making enjoyment, and look at how it relates both phenotypically but also genetically to social and well-being outcomes. So my first goal for today is going to talk to you a little bit about how we characterize the genetic architecture of social music making enjoyment. And then we next kind of delve into, okay, now that we have this kind of map, how can we, what are the ties with kind of social and emotional well-being outcomes that we can find? So as part of our work, we have kind of a highly collaborative lab, as you can imagine, for this kind of work. And so we partnered with a cohort called Lifelines, which is a multi-generational study. They actually allow for follow-up studies. So we basically sent to them a survey, including Barcelona Music Reward questionnaire items. And one of the questions that we focused on was this one: I like to sing or play an instrument with other people. So this question, as you can imagine, kind of captures social music making enjoyment. It's kind of this reward that you have when you engage actively in social musical activities. Participants could respond to 5 different options, from completely agree to completely disagree. And you can see that the distribution of the data is relatively normal. So that's kind of representative of what we would see in the general population. This was collected in over 30,000 individuals. And then before delving into the genetics, we actually looked at, okay, can social music making enjoyment predict your life satisfaction in a way, different facets of life satisfaction as kind of a broad measure of well-being. And so what we found is that over these 5 different items that capture life satisfaction, one of them, for example, is literally asking, okay, are you satisfied with your life? I mean, those measures are kind of approximate, but they're interesting. What was really interesting to see here is that our social music making enjoyment variable was a predictive of all different life satisfaction measures, including after accounting for different covariates that we know are associated with life satisfaction, such as income, education level, age, etc. And so now, kind of trying to look from the genetics lens, what we found here was that social music making enjoyment is also heritable. At least in the Lifelines cohort, we looked at how much genetic variation would explain social music making enjoyment, kind of individual differences, and found that there's actually a SNP-based heritability of 12%, which is, I would say, interesting for just a single item, comparable to what we would see for other behavioral traits, including some traits such as like ADHD or autism spectrum disorder, for example. Okay, so now how did I proceed it to kind of characterize this genetic architecture of social music making enjoyment? Still using Lifelines, I recombined 3 different batches of genotyped individuals, conducted a GWAS, meta-analyzed the kind of like summary statistics, and then kind of got this final Manhattan plot on over 19,000 individuals for this single item. And so, as you can see, we're all very sad. There are no genome-wide significant hits. Sorry, meant to go back. Okay. And so, but we can still find some, interesting signal, LD patterns. So I think that it's more, we should take it as this is promising signal. We still need to ramp up the numbers, as we can expect. 20,000, almost 20,000 is not really enough to get robust genetic signal. But what is interesting, I think, to ask is also, okay, although we do not have genome-wide significant hits, can we find some evidence that this GWAS signal is valid and tells us something about human musicality and not just, random kind of random noise? And so what we did was correlating social music making enjoyment GWAS with previous GWAS of musical beat synchronization, which was done by our lab 3 years ago. And we found a correlation of 0.6, which I think is pretty significant, as musical rhythm is definitely a skill that you expect to be involved in as you engage socially in musical activities. To be able to sing with others, to be able to play an instrument, you definitely need a kind of a sense of rhythm. So it was interesting to see that it was genetically correlated. We also found that in BioView, we managed previously to identify musicians through the medical notes. So we kind of searched for keywords like guitar, piano, etc., and managed to identify a set of cases through their electronic health records. And so what we found is that if you compare cases for musicianship versus controls, we actually found that the polygenic score for social music making enjoyment, so your kind of genetic predisposition for social music making enjoyment, actually predicts whether you are a musician or not in the direction that we expect, which is really interesting as we're trying to figure out how to validate the signal. So our next step was to look at genetic correlations, kind of exploratory, between social music making enjoyment and different personality, psychiatry, and social connection traits. We found 2 significant correlations. So social music making enjoyment was associated genetically with openness and extroversion, which are 2 not only personality traits, but they're also socially relevant personality traits in the sense that they will drive some of your social behaviors. And we also found, as you can see, kind of some trends for like loneliness and other traits, but those did not, unfortunately, pass multiple testing correction. But as it relates to loneliness, I kind of managed to get, this is a little bit small, sorry. But essentially, the main message of this is that in all of us, I managed to use the data that was derived from the social determinants of health surveys that they have available. And I basically derived polygenic scores of social music enjoyment in the All of Us data that with the social determinant of health survey, and managed to look at, okay, can your genetic predisposition for social music making enjoyment predict your loneliness status in all of us, above and beyond your genetic predisposition for loneliness? And what we actually find is that this is true. So the higher your genetic predisposition for social music making enjoyment, the lower your loneliness scores, the higher your social connection scores, and that's above and beyond your genetic predisposition for loneliness. So I think that's kind of a proof of concept that was found for 2 items of the UCLA loneliness scale out of 4. So I think what was really interesting looking at those findings is that maybe there is something going on in the biology where your social music making enjoyment actually may be counteracting part of your loneliness, or can help, let's say, overcome some of your genetic predisposition for loneliness. I think this is even more relevant when we have in mind that 3 or 2 years ago, actually, the US Surgeon General actually kind of released a report on the kind of what we call the epidemic of loneliness that is currently going on in America, but I think also in many other countries. And so I think that looking at how social music making interacts with loneliness potentially also at the biological level may be relevant to address this kind of really big and growing problem we have. So some key takeaways: social music making enjoyment is heritable. That was one of the main kind of findings that we have from lifelines. We then looked at some genetic correlations and found associations between social music making enjoyment and different personality traits, openness and extroversion specifically. And then we also found some interesting not trends actually, I mean an interesting signal between social music making enjoyment and different aspects of well-being, from life satisfaction to loneliness and social connection. So some of the future directions, of course, there's a critical need for enhancing the diversity of the samples. Currently, musical phenotypes are mostly collected in European data sets. I mean, we're already having this problem with other data that are being collected for medical purposes, for example. So you can imagine, but for musicality is a bit rarer to be studied. And therefore requires definitely some effort. And our lab is trying to collaborate with diverse samples to address that specifically. Next steps would involve also kind of looking at evolutionary function of social music making, and then kind of identify potentially some biological role of social music making through some enrichment analysis that I'm currently working on. So with that in mind, thank you so much for being here. And here, okay. Would like to thank my lab, the Vanderbilt Music Cognition Lab, all my external collaborators from the Max Planck Institute for Psycholinguistics, my committee members, and then our funding sources, of course. And I'll be happy to take any questions.
Any questions?
That was a fun talk. I feel like most kids are really young music. I wonder if you could use with kids as you positive control signal. That our genes are not stable, so I guess loneliness.
Yeah, no, that's a really cool question, and I think, I mean, I have also very much an interest in looking at like children musicality. The question that is a bit hard right now is that we really do not have much data on children specifically with the musicality aspect, and like usually what most measures that are being captured are parent reported. So parents are going to kind of tell you how much they think their child engages in musical activities. Either it can be like once a week, twice a week, or several times a month, etc. And so those variables are a bit complicated to work with, but we've tried to kind of use those as a starting point. And so yeah, that's a really valid question. I'm personally really excited to kind of start integrating all of this together.
Yeah, my question is about stratifying the population that you're sampling.
Okay.
Because there are several degrees of people enjoying music.
Mm-hmm.
Those that just enjoy listening to pop music,
Absolutely.
versus those that play an instrument and have a career out of it.
Mm-hmm.
Are you planning to actually take a deeper dive into what it means to enjoy music?
Yeah, so that's a really good question. I think there is some not conflicting, but in a way there's different views. I think that so far most of the research is focused on actually creating a gap between individuals who do not kind of engage in music activities and then individuals who are very accomplished and go for a musical career, or they're musical students, etc. And as a kind of we as a lab are trying to kind of pitch actually musicality as something less stratified and something that actually engages with your whole population. So we're trying to say individuals are going to, they may not play an instrument, but they're going to potentially sing, even if it's in the shower, which, I'm sure some of us do that. I think that there's a way that, just listening to music is being musical, or just enjoying to go to karaoke with friends, even if we're not like expert vocalists, is going to be also being musical. So I think our lab actually has tried to kind of move away from the purely musicians versus non-musicians kind of paradigm. However, it is very valid point that musicians, of course, have maybe also at the brain level, for example, certain neuroplastic changes that are very worth investigating. There's also new, like we see that in FIWAS approaches, for example, where there's also new clinical outcomes that arise for musicians, not necessarily because music is bad for you, but probably because if music is taken too far in a way, or like you're engaged in music formally, then you become potentially a bit more anxious because that's just part of your profession, etc. So I think there's all these different layers, and I do agree with you that that's worth looking into. Maybe as we ramp up sample sizes, that will give us more power to do so too.
Thank you very much. I have a million questions, but I'm looking at the clock. So
yeah, absolutely. You're really good. Thank you so much.
Okay, now we have our last abstract selected talk from Sayumi York from Notre Dame of Maryland University. Just the big button.
Hi, everyone. Thank you for coming to my talk. Today, I'm going to just give you a brief heads up on what's going to happen in the next 15 minutes or so. I'm going to talk a little bit about my organization, Seymour, our collaborators, our curriculum, and our overall goals and challenges of what we're trying to accomplish. I'm then going to talk about some of the current features that our curriculum has, as well as some of the infrastructure that we're currently using on AnVIL to support it on the platform. And finally, I'm going to talk about some challenges that we've identified in the last 6 months that I'd like the community to think about as we move forward to develop the AnVIL platform, if we're going to intend it for more widespread adoption for educational use. So first, a little bit about myself. My name is Sayumi York. I'm the Genomic Data Science Educational Developer at Seymour. And while I'm officially headquartered at Notre of Maryland University, we are a cross-institutional, cross-organizational group of people who all collaborate on this project. You can see some familiar faces in this picture here, but all the way from the East to the West Coast, a lot of different people from computational backgrounds, instructor backgrounds have all worked on this. So I'm happy to speak not only for myself today, but to represent these people and these organizations, as well as a shout out to some of our other collaborators that we work with. So we at Notre Dame are part of this group of universities that we call ourselves as the UE5 sites, because that's the name of the grant that all of us were fortunate enough to receive to create educational materials for cloud platforms like All of Us or AnVIL. I also want to give a shout out to our collaborators at Grads4C and the Genome Data Science Community Network, who we've been working closely with, especially as we develop a biodigs curriculum. So again, there's lots of great work in this area. We're all tackling this issue of education in the cloud in many different ways. So it'll be interesting to see how all of us move forward in these next few years. If you're in the audience here, you're probably already interested in the cloud, but just in case you aren't, let's take a look at the numbers. This is a 2017 study on NSF PIs in biology, not including bioinformatics, about their present needs in 2017, and then they predicted their needs again for what they would think of as a need in 2020. And I've selected only these 5 items from the survey because these are the 5 items that our curriculum are trying to address: bioinformatics analysis, basic computing and scripting, data set discovery, multi-step analysis, and of course, cloud computing. And you can see in all cases that their current need in 2017 was lower than in the future 2020, their predicted need at the very least. And we understand how dramatically the landscape has changed in the past couple of years. So you can imagine that now that need is even greater with the forecast of the rise of AI, increasing sequencing technologies, and the need for greater collaboration. And so these skills are sorely needed. What we specifically at Seymour are trying to address is making sure that these educational opportunities are equitable for all, regardless of what kind of background you come from or how many resources your institution has. Another thing that I'm going to need to talk about today is that we are teaching based on this CURE model of education. And CURE stands for Course-Based Undergraduate Research Experience. In a traditional lab, students will do experiments where the instructor kind of already knows what's going to happen, and oftentimes so does the student. Students are modeling some kind of natural phenomenon empirically in order to demonstrate its use in the real world. So I come from a marine ecology background, and so one of the labs that we would have students do classically is a digital lab where they remove organisms from a tide pool environment and see how that affects the community structure. In a CURE, however, what you would do is you would actually take your students out to the tide pools and have them do real research on a question that they themselves formulate. So it's a really interesting model because no one knows the outcome of the experiment, right? They're doing real, genuine research. And not only are they modeling some kind of phenomenon, but they're also modeling the scientific process in itself. And so they're building these really critical skills about hypothesis making. They are practicing their critical thinking skills when they have to troubleshoot. And what's really great about this is that it really opens up the field of research, especially for students who might not previously come from a background where they know that research opportunities are available to them. And it can change lives. I know because I'm one of these students, I didn't know that research was even an option for me. And so it's a wonderful thing to be able to experience. Obviously, not everyone teaches a cure. And part of that is because cures are incredibly demanding on the instructor, both in thinking about how to get logistics working properly, students from place to place. There's a lot of extra time and finances that are built into it. And so this data is from a 2015 study on instructors at universities who are thinking about implementing a cure, but these are some of the challenges or hesitations they have when they're developing their course. And so we wanted to be able to have the fulfilling experience of the cure, but we also wanted to reduce some of these hurdles in order for more instructors to get them working in the classroom. And at Seymour, we're addressing this challenge through the idea of what we call a mini cure, which is a compressed version of a regular cure. Traditionally, a full cure takes a whole semester or one block in a month to teach, but we are taking this computational approach. We have 6 modules that are designed to be integrated into pre-established classes. So the instructor only needs a fraction of the semester to teach it, and their load for creating the modules and deploying them is greatly reduced. For instructors that have no prior bioinformatics experience, we provide support to both them and our students through our online discussion forum. And then taking this computation approach also reduces fees on behalf of both the instructor and the student. So pretty much nowadays, every student has a smartphone, computer, or tablet, or they can borrow one from their institution to run our analyses on. They don't have to pay extra for a plane ticket or find someone to cover their part-time work. And so all of this really is to open up this computational curriculum for other people. And we've been doing this through the past 10 years. We currently have 4 different curricula that we've taught about 30 times. So here we started in 2015 from our initial co-founders at Clovis Community College and Johns Hopkins University. We started with the RNA-seq minicure. We then built that out into a single cell minicure, which is now going to be reintegrated. So we're very excited about that. We expanded to 2 more colleges. And then just in this past 2 years, we've developed those final 2 curricula. And again, we've expanded to more institutions and increased the network that we collaborate with. Those are the classes by the numbers. Here are the students. The vast majority of our students are first-year students. We have taught over 700 of them. And we choose to have the research poster as their final deliverable. It's great for the students because they can have something they can physically look at, they can take it home, they can send it as a PDF for future research opportunities. And when given the chance to present at research symposia or conferences, more than half of students choose to do so. One thing that we're really proud about is that we use these posters to actually start an annual STEM Research Symposium at Clovis Community College in 2022 that is still continuing to this day. And not only are students presenting at these conferences, but they are also winning awards. Students have participated not only at their local institution, but they've also participated in ASM meetings at the state level. We had a student present at Grads 4C. And our online summer group, we're happy to say that all 3 students had their abstracts accepted to Abrachems, and the 2 of those that were eligible for a travel award also received those. And so what we're trying to say here is that not only are we teaching a lot of students, but our curriculum is opening up new opportunities for them to grow their network, share their research. We're also publishing soon, I hope, our first micro publications. So that'll be our first peer-reviewed product that comes out of Seymour. We have 2 curricula that are going to be on AnVIL starting this semester. The first is our Drosophila RNA-seq minicure. In this minicure, students are looking at a published RNA-seq data set. So the data they're working with are very real. And they are going to look at the distribution of counts across the midgut and then relate those counts back to the specialized functions of the gut itself. We also have our brand new Human Gut 16S Microbiome Minicure. This one actually has 5 data sets from published studies to choose from. This is just an example of one here, which compares a high fiber to a fermented diet. And students will create a relative abundance graph like this, and they'll do other advanced analyses like alpha diversity, differential abundance, and PCOAs. And so the kind of research they're doing, and remember that these are mostly first-year students, is very high level, right? These are the kinds of analyses that you would also expect a grad student or a PhD student to be able to do in the lab. So it's very genuine, very real, and very exciting. And what we've done with these two mini cures is we have built them into their own workspace. And these workspaces are public, so you can actually go ahead and clone them right now and try them out for yourselves if you like. But what we have is each student clones the workspace, and then all of the materials that are needed for them to actually launch the module that don't come by default on AnVIL, we've loaded into this startup script. So those are all of our specialized R packages, and then the RMD files themselves are stored in GitHub. And students use that startup script to then open the RStudio environment, and then they launch their Markdown file from there. And so what it actually looks like in use to the student is something like this. It's a very friendly, almost textbook-like appearance. They have a table of contents on one side that allows them to jump through different sections of the textbook, take code from different analyses. We have instructions for them. They have their own interactive code chunk that then prints out all of their plots and tables within the module themselves. So they'll just screenshot those graphs to put them into their poster. And then we have what are known as quizzes, or I like to think of as knowledge checks, where we ask a question to the student and they can take this quiz any number of times in order to refine their understanding about what's happening at the molecular level, or test their understanding on what the results mean. Excuse me. And even though right now we are creating this curriculum to be deployed synchronously in-person classes, we've set it up so that in the future, this could facilitate the use of the curriculum in an asynchronous or online only platform. So students are really, in this case, they're able to control the pace of their learning and their access. Some of the problems that have cropped up within the next 6 months that need to be addressed before AnVIL can be released as an educational platform to the public as a whole. Instructors need more transparent and flexible billing options. If you have taught a class recently, you know that students, they'll forget to put away their physical lab materials. Well, that's doubly true more so for their computational lab materials. There's human error element and all of that, right? And there's a lot of fear and apprehension from new instructors when you tell them that you could continue to have runaway costs if things aren't shut down properly. So ideally, we need to be able to measure the spending at the level of individual student and also give instructors the ability to toggle their access on or off. Another thing that's really big right now is the startup time of the RStudio environment is very long compared to the amount of time we have in the classroom. Our current tests have RStudio starting up anywhere from 5 minutes on a really good day to about 15 minutes, maybe around 12 minutes on average. And this is really restrictive because when you think about it, some of your lab classes are only 90 minutes long. And we can kind of counteract this by having the instructor lecture or discuss with students while AnVIL is loading. But it's difficult to get around that, especially when you're not sure where in that 15-minute window you're going to land. And you can imagine that right now, the instructor is designing their lesson around AnVIL rather than being able to use it as a tool that they can use freely. Finally, we need to create more onboarding and continued training support for instructors. The platform needs to be accessible for instructors who are not familiar with cloud computing or genomics. Even someone like myself who has previous omics experience but has never worked on a platform like AnVIL before, some of these terms like workspaces and billing projects are very esoteric. In particular, we found that in our experience, billing setup can be really complex. Not only do instructors have to understand the difference between billing project and Google Billing account, but setting up the account can involve multiple departments at the university, whether it's accounting or IT, and it's much more complicated than the sign up that they might used to do, like just signing up for logging in on Galaxy. One other thing on there that's really important that I wanted to mention today is at Seymour, our philosophy is to always provide instructors access to all of the older materials. So for example, something that's happened within the just last 72 hours for us is we were all ready to start the new semester, and then we logged into AnVIL and we found that Bioconductor 2.19 was no longer available at the start. And we went, oh no, because our curriculum doesn't work on 3.20 and above. And so we had to find a workaround for this, but it was really kind of a shock to suddenly lose access to something that we knew worked and then have to reroute it. So I know that the continuing evolving of bioconductor is really important for the researching community, but being able to have those older materials is really important for instructors, because once we have something working, we don't want to be booted off it without notice. We want to be able to choose when we have revised our curriculum. And so with that, I'd like to end my presentation. I'd like to thank all of the wonderful people I've worked with, as well as all of our collaborators and funding sources. I can take some questions now. Thank you very much.
Thank you. I think we can all agree that impact is enormous with 700 students. I also really appreciate the critical feedback on some. Any questions from the audience?
Wonderful work. I really appreciate you putting the educational aspect to this, because neurokids, if they own a certain platform, that will stay for a longer time than us. And then I also want to know whether you have some plan for people like me, old ages, and we need a refresher on the new technologies. You know, like I know Unix, but now we are on the cloud. And then I have to go search on YouTube, and there are a bunch of like people say, this is how you do it. But sometimes the video is really helpful because I only need to see, oh, this is their data set. If that's a step we need to do, how long finish, and that give you a sense of, is that approach I can take? Or sometimes I say, oh, that's not going to work for my data. So, but I really feel there's a gap. If now I have to learn how to use Word, Excel, because what I get used to do 10 years ago is not going to be very efficient for me anymore. So that will be a good aspect.
So the question is really about how do we get instructors more training, especially when they are maybe technologically less native. And right now, the way we have it set up is that we really encourage instructors to come speak to our organization at the level of individual instructors, because we can provide that kind of personalized support. I'll say that we have one new instructor this semester that is not really familiar with computing at all. And we've helped her set up her account. We've set up her workspaces for her, and we're in constant communication with her on any problems that might happen moving forward. In the future, I believe that our hope is that we can create some kind of training model where we have a summer team, perhaps, of instructors that work together and teach people how to use these different materials at their institutions during the school year, which is more intensive training then. And then during the regular semester, we're still there as a resource for them. I know that we are actively looking for funding opportunities right now to build that training hub. I hope that answered your question.
We have a question here.
There's a question from the Zoom chat. This is a question from the Zoom chat. How many students in a class were trained to use AnVIL?
So the question is, how many students per class are trained on AnVIL? Right now, we have student class sizes of anywhere between 2 to, I believe, 25 or so in a classroom at a time. So that's per section. So some instructors, I believe at Clovis Community College, we have multiple sessions. So she's probably teaching maybe 50 students at a time there, but not all synchronously.
All right, thank you so much. And let's thank all of the presenters.